pair. So if there is a A gene here, it should contain another A gene here. If there is a B gene here, here is a B gene. If here is a C gene, it is C gene. They are homologous, identical chromosomes. So these are called homologous chromosomes. Okay. So when we will talk about this genetics part, we need to think about this homologous chromosomes. So each and every chromosome pair, chromosomes, that is gene containing chromosomes, they have a pair. Okay. Suppose now in each pair I have these genes. This is the homologous chromosome pair. Okay. Now there is one chromosome and in another parent there is another chromosome. Same chromosome. Think about it. Or make it small. Okay. What will happen during meiosis? During meiosis, these two chromosomes will separate and produce two gametes, sperms or ova. Okay. Any kind of genetic cell, gametic cell, which is haploid, the first term is called haploid. Then if it is haploid, so what is the other term? The other term is called diploid. Diploid means di. This is actually, di means two. So these are diploid chromosomes, diploid cells, 2n. This will produce two gametes, each will contain the chromosomes separately. Okay, this is after meiosis. And the stage of meiosis are in meiotic first division, these two homologous chromosomes separate into two cells. During meiotic two division, from each homologous chromosome, two chromatids separate. This is the main part, main summary of this. Okay, meiosis. So these two types of gametes will produce. From here, another two types of gametes will produce. Clear? I have started from very basic things. Okay. Now, when they will mate, each can produce, again there will be 2n cells. So these things we can draw by this way. So from this parent, if it is male, this is the symbol of male. If it is female, this is the symbol of female. Okay? And these are all gametes. I like the term gametes because you will find in your book, that is the Campbell's book, you will find sperm and ova. But sperm and ova is only applicable for animals, not for plants. For plants, there are pollen, gametes, and so many things. Pollen, ova, all those things. So different kind of gametic cells are there. So I will suggest just write gametes, male gametes and female gametes. Wherever it is sperm ova, wherever it is pollen ova, that doesn't matter to you. Okay? So these are called gametes. So this, if we will draw like this way, this type of square is called Punnett square. Punnett square, I think double T. So here, what we will do, we'll separate the gametes first. So from this male, there are two gametes, A and A. This is male, there are two gametes. This is female, there are two gametes. So these are all gametes. Two gametes will produce one diploid offspring. So this is called offspring. Okay. So these gametes will produce, so from here it will be A, here it will be A. From here, like this. So there will be 4 types of offsprings can be produced from here. Okay? Clear this part? Now, in Mendelian genetics, Mendel worked with 2 characters in pea plants. So he took 2 characters, and those characters are, so pea plant, there is purple flower and white flower, but I will take another 2 things, the height of plants. This is called character, the height of plant. Now, if it is the character of the plant, there could be 2 traits. Okay, so this is the character. Now the traits are either it could be tall or it could be short. Okay, so there are 2 characters. Either it could be, there will 2 traits, that is either tall or short. And now these 2 things, what Mendel did, he took 2 plants in diploid condition. So this is pure tall plant and this is short plant. I am taking 2 genes, names like this, T and small t. Okay? So this is in diploid condition. If this is diploid, so they must have 2 capital T here and they must have 2 small t here. Okay? When they crossed, or he crossed these 2 things, he got a plant which is tall. But according to this scenario, they can produce one type of gamete, that is small capital T, and this can produce another type of gamete, that is small t. So this plant must have this type of gametic condition. Clear? Any problem, question? I think you already read this part in high school. So this is tall character. Now, there are 2 gametes here, but the character is tall, the trait is tall. From here, he concluded one part. Next, when he crossed these 2 plants, what happened? So, there is, I can draw the Punnett square here. Male, female. So from male we'll have these 2 characters. From female we'll have these 2 characters. So what will be their outside characters? Yes.
So most of them will be tall, but some of them will be short.
Which one is tall and which one is short?
The short one's the only one that has a TT, and the rest will be tall.
Which one? This is short.
No, the small low
This
piece.
is short. Now, from this one, so we are thinking about 2 things. One is their genetic combination, another is their outside character. Genetic combination is called genotype. Outside combination, that is the character, that is called phenotype. Can be pronounced in several ways: genotype, genotype, phenotype, or whatever it is. But these are the things. So we have the genotype, capital T, but the phenotype is tall. Clear? So phenotype is tall, phenotype tall, phenotype tall, but we have the genotypes like this. Now, if we think about the phenotypic ratio, what is the ratio of the phenotype? So we have 2 characters only, tall is to short, we have 1, 2, 3 tall characters versus we have one short character, right? That's fine. What is the genotypic ratio? Genotypic ratio means we have three types of genotypes here, which is 1 is to 2 is to 1, right? Now, based on this experiment, what Mendel concluded, the first thing is that there are two traits, he got two traits during the first generation, which is called first filial generation or F1. During first filial generation, he got just tall plants. So that means the tall character suppressed, not suppressed, probably took out the short character here because we couldn't find anything, any short character, right? Now, in the second generation, this is called second filial generation or F2. In F2 generation, we got short character again. That means it was suppressed by the tall character. It was there, it was suppressed, but is expressed only when both small t are there, right? So due to this result, he concluded that one character is dominant over another. So here the tall character is dominant character and short character is recessive character. So the concept of dominant and recessive. Now, if this is in pure form with the pure two genes, this is called homozygote or homozygous condition. Homo means same. Homozygote or homozygous condition. If so, these are both are homozygote or homozygous. But these one have both genes together. This is called heterozygous. Hetero means different, zygous. When it is in heterozygous condition, that means the dominant gene and recessive genes present together. The dominant will express as phenotype. Okay? So we got two concepts. One is dominant and recessiveness, another is heterozygous and homozygous. Now, after this test, we got the ratio, phenotypic ratio, that is tall versus short, 3:1. Genotypic ratio is 1:2:1. This is called Mendel's monohybrid cross. Clear? This part. Why monohybrid? What does it mean? First, we need to know about what is mono, and the second thing is what is hybrid.
So it's one. So it means one hybrid. So hybrid is like a combination of.
Hybrid means the combination of two different traits. So that is the heterozygous one. That is called the hybrid. So this is called hybrid, and we got one hybrid for one character, that is mono. That's why it is called monohybrid cross. So what is another one? That is called dihybrid cross. So it is clear, like how the dominant is suppressing the recessive if there is a heterozygous condition. So I talked about a lot of things in one very short period of time, because when I am teaching this Mendel's class, it is almost like an hour. Okay, so the very in summary, there are two characters, one character, two traits. Character is length, height of the plant, traits tall and short. So this is monohybrid cross, as this is one character. We got homozygous and heterozygous concept here, and we have found the genotypic ratio and phenotypic ratio in FT generation is 3:1. Okay. Now, if I will tell you that we will do a dihybrid cross, can I do that? What is the dihybrid? Now, this is a little bit difficult, but not problematic. So dihybrid means we need to take two characters. First thing, don't mix the terms characters and trait. Sometimes we can say the character and trait together, but don't mix the term character and trait. Character means the character 1, we took yellow seed color. Character means seed color. Seed color. And character 2 is seed coat. There are two characters. Seed coat or seed shape, something like that. Seed coat or seed shape, you can say seed shape. So Mendel took two characters. There we took only one character, the height of the plant. Here we are taking two characters, seed color and seed coat. So each and every character must have two traits. Okay? So the traits. What are the traits? Seed color trait is either yellow or green. Okay? And seed coat or seed shape is either round or wrinkled. Now, if there are two traits, so we need to think about that which one is dominant, which one is recessive. Right? Here, yellow is dominant, this is dominant over green, recessive. And here, round is dominant over wrinkled, recessive. Generally, now we need to take the genes for those two. And so I think this is over, right? So how it is dihybrid? We need to take the pure forms. Generally, sometimes you can find a little bit mixing of characters, but I would like to take the pure forms. So the pure form is that these are two dominant characters. So the character is yellow round. One seed which is yellow and round. Another seed is green and ringed. Okay, this part. For this yellow character, we'll take the gene capital Y. For green character, generally, if these are two traits, we will not take two different names. So we are taking the character like here it is yellow. If yellow is dominant, for the green we will take small y. Okay. For round, it is R. So for wrinkled, it is smaller. Now, if it is the yellow round, what should be their genotype?
Uppercase Y and uppercase.
What?
So uppercase Y,
For yellow, the uppercase Y.
and then you put the uppercase R for the round part.
So like this. Like this,
Well, together,
both together.
and then the other one would be lowercase Y and uppercase R, and then
Are you sure? Are you sure?
if I'm remembering correctly.
Okay.
Y-Y-R-R.
I'm going for that one. So we have a gene here, a chromosome here, which contains Y, and here is a chromosome which contains RG. Right? Is this okay? Okay? Everybody? Why not? If you say okay, you need to say why. If you say it is wrong, you need to say why. Okay, why is there always? Why? Two. Why? They could be pairs. So they must have homologous chromosomes. If these are homologous chromosomes, they should have this in this format, right? So the genotype would be, make it clear. Okay? So the genotype would be YYRR.
Oh,
Clear? If you cannot face the problem, you cannot understand. So I am always telling you, just give some answer. Whether it is wrong or right, that doesn't matter. But you can understand, right? This was your problem. Now you can understand. You'll never forget this one. So if this is true, what will be for this one? Right? Okay. Just remember this one. Now, if we have a gamete from here, what should be the gamete? I'll ask him. No, Alex. What should be the gamete? What should be the gamete? Anybody? These are in one cell.
yeah.
YR. Is he correct? Or not?
Yeah, because it's going to be only half of the.
So YR in one cell or Y one cell R one cell?
Why are you watching?
YR in one cell. Why?
Because instead of having the two, it's just the one. You're just looking at the one, the two, one.
So the homologous chromosome will separate
Yeah.
in such a way. So homologous chromosome will separate during meiosis. So it would be like YR. This is the gamete. And from here, the gamete is this. Right? If these two gamete fuse, what will happen? We will get this combination, right? This is a hybrid, having both dominant and recessive characters. What would be the phenotype?
Yellow round.
What would be the phenotype?
Yellow round.
Are you sure?
Mhm.
Why?
Because the dominant traits are there.
Dominant traits will express. So it will be yellow and round. Okay? Now, if I will self-pollinate these genes, what will happen? Now, this is a very big table.
No. Let's see.
So I will self-pollinate these two characters. The same thing twice.
And it's fair. Easy.
What would be the male gametes?
Big.
It would just be like either YR or YL. Like uppercase YR. And then lowercase YR.
Lowercase y.
And then you have the Y with lowercase R and then lowercase Y with uppercase
So
R.
we have this one also,
Yeah, and then
and we have this one also. Right? Right? Everybody agree? Now, what will be these two? phenotype.
So it's BYR.
This phenotype, right? This one I have 2 capital Y, 1 capital R, 1 small.
Y, capital R, focus on.
No,
Why
it's BYR because two dominant states.
So it
So
will
the
be
one-
yellow round.
Yeah,
Yeah,
but
yellow.
Yellow round or yellow gray?
Yellow wound.
But this one,
It'll still be yellow wound.
this one, yellow round. Okay, here, Y, yellow round, Y.
Yellow green. No, not yellow. Yellow
I mean,
wiggle.
yellow wrinkle.
Wrinkle.
Yellow, link.
Yellow round.
Yellow round. Here,
Yellow wrinkle. Yellow round.
Yellow wound.
Yellow round.
Green.
Green round. Green round. Round.
Yellow round.
Green round.
Green round. Green round.
green?
Wait, I wasn't thinking that like he was riding.
Green.
Oh, that's green
Wrinkle.
wrinkle.
If you think it was wide.
okay.
I know.
Now you have a very nice pattern here, right? Yellow round. Right? All are yellow round? How many are there?
7.
1, 2, 3, 4, 5, 6,
7, 8,
8, 9.
Come on, Andrew.
Oopsies.
Get a
Now
comfortable
I have this one, and this one.
3.
Yellow wrinkle. 3. Now here, same thing. Green round. 3 and 1. Green wrinkle. This is Mendel's dihybrid ratio. 9:3:3:1. Okay? If you do the gametes in this way, you will get very good combination, just like these triangles. Okay? If you mix it up, then it is gone. So this is the Mendel's dihybrid cross ratio in F2 generation. In the first cross, monohybrid 3:1, second cross, dihybrid cross 9:3:1. Okay, now I have some corns here. One corn. Okay, before that. Now, think about the monohybrid cross again. So in the monohybrid cross, we got this combination as a hybrid one. Now, if I cross this one with the recessive one, what will happen? And if I cross this one with the dominant one, what will happen?
So with the dominant one, it would just
Because this phenotype is tall, so we don't know whether it is heterozygous or homozygous. We don't know if we don't know the gene. And at that time, there was no way during Mendel's time in 1870, there was no way that he could do it by gene analysis. Okay. So this is tall and this is short. what will be the result from it? We'll get two types of gametes from this parent
Purple.
and one type of gamete from this parent, right? Tall and short. From here, what will you do?
So sure.
What is this? Tall. What is the difference between these two? So I took the that is the hybrid one. I crossed with the dominant one. I crossed with the recessive one, and got two types of results. So what can I conclude from here? Now what is the ratio?
So one's 1 to 1 and then one's just.
Here is the ratio is 1:1, tall versus short, and here is nothing. Everything is tall. Okay. So if there is a heterozygous plant here, if we cross that plant with the recessive one, there is a chance that we can separate out the recessive character. If I cross this heterozygous with the dominant one, there is no way we can separate out the recessive character. During Mendel's time, or 1800, 1870 or something like that at that time, or 1900, there was no way that you can tag with the genetic probe to find out the particular gene or the telomeric region, not like that, FISH technique or those things. So he did these crosses to find out whether this is heterozygous or not. So this cross can tell us whether it is heterozygous or not. As it is telling us whether it is heterozygous or not, that means we are testing the heterozygosity with this cross. Each and every word is important. We are testing heterozygosity by this cross. That's why this is called test cross. If it is a test cross, the ratio should be 1:1. Clear? Any question? Now we have couple of plants, corns. In these corns, we have two characters. One is purple, these are all purple, and yellow characters. Okay? So what you need to do, in one row, you need to count how many purple are there, how many yellow are there. I would suggest to count at least 3 rows. If it is yellow or purple, that means there are 2 traits, the color of the seed, traits are yellow and traits are purple, and this is a hybrid plant. So what ratio we would expect? Monohybrid cross, this is 2 characters. Monohybrid cross, that is 2 traits for 1 character. Monohybrid cross, we would expect 3:1 ratio here, right? You need to check whether there is 3:1 or not. So I would suggest not do the just side by side. Take 3 random rows, count the number of purple colors and number of yellow colors, and find out that whether it is 3:1 ratio or not. Okay? So this is your first experiment. Okay. This one is first. Don't mix up these things. Okay? Now the second one is that we have these plants and there are 2 characters. Come closer so I can show you. So this one is called wrinkle character or shrunken, this part, and others are round. Okay? And there is purple and yellow. So we have 4 traits and 2 characters. The round shaped seed or wrinkle shaped seed and the 2 colors purple and yellow. So what is this?
So
Trihybrid or trihybrid or pentahybrid, hexahybrid,
It would be, so there's two traits, so it'll be a dihybrid.
dihybrid cross. So what ratio we could expect from here?
Seven,
1 to
rather
1,
nine to
2 to 1, 3 to no.
9, 3, 3.
9:3:2, 3:1. So you need to find out in this way. Same. Take at random 3 rows, count this, note it down, and then find that whether there is 9:3:3:1 ratio or not. Okay? So this is for dihybrid. Don't mix these things, otherwise it will be mess. And from here, we'll find this is. I think this is mixed. Okay, do those things fast. Actually, we will get only one is here. So there is yellow and purple color only. So we need to find out whether there is. This is a test cross. So what can we find? Ratio. This is actually dihybrid trace cross. You need to draw that. So I showed you that monohybrid trace cross. You need to do the dihybrid trace cross. So count from here, and you will find 1:1:1:1 ratio. The same character, dihybrid character is there, and this one is the same character with wrinkle seed, round shaped, and 2 colors, yellow and purple. Here we have just 2 colors, yellow and purple for the monohybrid cross. For the test cross, we have the 2 colors and 2 shapes. Okay? So this is your first part. Not first part, the second one. This one. So do it.